Hello, everyone. Today, I'm going to share with you all some work that my lab has been doing on focal non-thermal ablation of brain tumors. And this is largely motivated by the challenges of successful thermal ablation inside the brain using transcranial ultrasound. Major hurdles, of course, are absorption by the skull of the ultrasound beam, which can lead to surface heating and thermal damage, particularly in the cortex. And this is due to the use of continuous wave ultrasound, which is predominantly used for thermal ablation of solid tumors, as well as the very high powers that are used that can be on the order of 1,000 watts per and greater per centimeter squared. So a big question is, can we use cavitation in order to reduce the acoustic power and also enable non-thermal ablation inside the brain? And can we potentially increase the localization of the precision which we are able to achieve this non-thermal ablation? So in our lab, we've been using phase shift nanoemulsions, which are basically ultrasound contrast agents or fluorocarbon microbubbles, which have been condensed down to a submicron size. These do have a lipid shell, which does prolong circulation upon systemic administration. And the lipid shell also can improve the stability of the particles. Ultrasound is used to vaporize these liquid perfluorocarbon droplets in order to reconstitute or form the perfluorocarbon gas bubbles. And those then are driven to achieve cavitation. For the work that we've been doing, we've been using inertial cavitation, so these collapsing bubbles, in order to facilitate non-thermal ablation. Compared to microbubble facilitated ablation, or using the microbubbles for cavitation, we can spatially control the vaporization of these perfluorocarbon droplets, liquid droplets, such that the cavitation field is highly localized to the focal spot of the transducer. That does allow us to avoid damage in the intervening tissue between the transducer and the focal spot for the transducer. These perfluorocarbon droplets also can be injected at a much higher concentration than microbubbles, so we can achieve the concentrations on the order of 10 to the 9th droplets per milliliter, compared to what is typically used 10 to the 5th, 10 to the 6th microbubbles per milliliter for cavitation that is induced or nucleated by microbubbles. So the testable hypothesis here was we can use these perfluorobutane phase shift nanoemulsions, or PSNE as we refer to them in the lab, to improve the spatial control of cavitation-mediated non-thermal ablation of brain tumors, and thus avoiding prefocal damage. And the methods for the study were the use of transcranial focused ultrasound to activate these PSNE, as well as microbubbles, and then quantify and compare ablation size that was generated by inertial cavitation using MRI and H&E staining, so histological analysis. This is what the system or the setup looks like for the experiments. We did use 2 focused transducers that operated at 837 kilohertz, and then had a single element transducer that served as our passive cavitation detector, just to monitor the cavitation activity, just to confirm that we successfully vaporized the droplets. And we used tone bursts, 10 millisecond tone bursts, and of course had a pulse repetition frequency of about 1 hertz for these experiments. So the ultrasound beam propagation is shown at the very top through the brain. The cortical region is at the very top. The focal region positioned at a location within the brain. We conducted our early experiments with mice that actually we did not have established, rats, in which we did not establish brain tumors, just to make some comparisons, some initial comparisons between the ablation size for microbubbles compared to the fluorocarbon PSNE. And so this slide down here shows the MR images, which actually do detect the edema that's formed due to the ablation. In the cases where you have microbubbles, which is the top row, compared to the PSNE, which is the bottom row, the focal region, you have successful ablation and edema in both cases. When using the microbubbles, we also did detect or image edema in the cortical or in the cortex region compared to the PSNE, where we did not detect any ablation or edema in that region. The size of the ablation or the edema volume was much larger using the microbubbles compared to ultrasound contrast agents compared to the PSNE. And we quantified that for different concentrations from the stock solutions that we prepared. So 30% or 5% of the stock solution for the microbubbles compared to 30% or 5% for the nanoemulsions. And in cases comparing the nanoemulsions to the microbubbles of comparable dilutions, we see significantly smaller lesion volumes or edema volumes for the nano droplets compared to the microbubbles. This gave us confidence that we could achieve spatial control of the ablation zone using the nano droplets compared to the microbubbles. So the next question then, or the next part of the study, was to establish the brain tumors. In this case, we used F98 rat glioma cell lines to establish these tumors in Fisher rats, in the brain of Fisher rats, and then administer microbubbles, in this case, ultrasound contrast agents, or PSNE systemically, which then served as cavitation nuclei for local non-thermal ablation of these tumors. Once again, on the left here, you've got the MR images just showing where we had edema accumulation, and which was indicative of successful ablation. In this right panel here, we're looking at histological slides. And so for the microbubbles, we do see some damage on the periphery. It's largely limited to the periphery for these tumors. The dark gray, or purple, is the cancer cells. And if you look to the right column, you see the absence, basically, of tumor cells, which suggests that we were able to actually emulsify the tumors. And so you end up with this sort of region that's been completely ablated using these nanodroplets as the cavitation nuclei. In the periphery, we also still have some damage, but we see some residual cancer cells. And I'm going to come back to that in just a moment. But the percentage of the ablated volume using the nanoparticles, or the PSNE, compared to the microbubbles was much, much larger. So we were able to successfully ablate on the order of 85 to 90% on average of the tumor using these PSNE for nucleating the cavitation compared to the microbubbles. So the tumors were monitored over a couple of weeks after ablation. We ablated the tumors, or used ultrasound to ablate the tumors, nucleated with cavitation nucleated by the PSNE on day 8. And compared to controls, so no treatment whatsoever, we see a region of necrosis as well as ablation, but also continued tumor growth. And the tumors grew to a much larger size when we measured on day 24. The tumors were much larger as expected for the controls, but we do still see tumor progression even in the cases of ablation, non-thermal ablation, using cavitation nucleated by the PSNE. So in summary, we were successful with vaporizing these droplets inside the brain, achieving tight control, spatial control over cavitation using the PSNE compared to the ultrasound contrast agents. Ablation was highly localized and correlated spatially with the transducer focus when using PSNE to nucleate the cavitation, whereas we did detect prefocal ablation when US ultrasound contrast agents were used as the cavitation nuclei. The increase in animal survival after cavitation-mediated non-thermal ablation of the brain tumors was insignificant, and this was largely due to incomplete ablation of the cancer cells. And so more than likely, adjuvant treatment will be needed to eradicate those surviving cancer cells and improve the survival rate for these in these preclinical studies. I'd like to acknowledge the team members in this project, Cheng-Wang Pang, Jason Papademetrio, and the Focused Ultrasound Lab. It's led by Nathan McDonald, and the funding sources are from NIH and the Focused Ultrasound Surgery Foundation. And thank you for your attention.